In the late 1990s and early 2000s, the public and scientific communities were captivated by a single, powerful idea that human DNA was the ultimate blueprint of life. Leading scientists like Francis Collins, who headed the National Institutes of Health's genome sequencing efforts, made explicit promises. They claimed that decoding this genome would cure humanity's biggest scourges, like cancer and heart disease, within 10 years. 2 decades have passed since the first full genome sequencing. Those sweeping cures never materialized. This simple chart highlights the reality of what genetic sequencing actually predicts. It effectively targets rare, single-gene anomalies, like cystic fibrosis, which affect roughly 5% of the human population. The other 95% suffer from complex, multifactorial diseases. 20 years of research failed to yield cures for the vast majority of patients. The data suggests the core biological assumption of the 20th century, that genes alone dictate human health, was incorrect. A recent massive study from University College London put the predictive power of genetic association scores to a strict statistical test. The researchers asked a straightforward question: does relying on genetic data accurately predict who will get a complex disease? The findings were stark. The genetic data yielded as many false predictions as accurate ones. If a conventional pharmaceutical drug went to clinical trials and produced an equal ratio of negative to positive outcomes, it would fail to get approved. Outside of those rare genetic anomalies, commercial genome sequencing cannot reliably tell you what disease you will ultimately die from. The sequence of your DNA fails to predict the outcome of your life. A database that cannot dictate an organism's future cannot be considered its primary control system. To understand why the sequence fails to predict our health, we have to look down at the molecular level. DNA is a long, inert thread made up of 4 basic nucleotide chemicals. As molecules, they simply do what chemistry dictates. They bind together automatically in specific pairs. They possess zero inherent conditional logic and cannot make decisions. The popular biological metaphor describes genes as possessing intent, or the capacity to be selfish and drive evolution. But a chemical thread without an organism attached has no agency. Even the foundational idea that DNA perfectly replicates itself is flawed. Left to its own devices, DNA cannot accurately copy itself. The process naturally produces hundreds of thousands of genetic errors. The replication requires intervention. The living cell must actively deploy specialized proteins to cut, paste, and chemically correct the errors along the DNA strand. The living system uses DNA as a passive chemical database, managing and interpreting the code according to the needs of the organism. We begin our search for the cell's actual command structures at the edge, the outer membrane, which is densely packed with specialized lipid structures and protein channels. Here is a biological paradox. If you search the 3 billion letters of the human genome, you will find absolutely no genes containing the coding to build these vital membrane structures. Every human inherits their complete, initial membrane system directly from their mother's egg cell, entirely independent of the DNA pairing process. This diagram shows the logistical hurdle of operating a cell. If we scale a single nucleotide up to the size of a golf ball, the nucleus becomes a tiny circle inside a massive country representing the whole cell. When a calcium signal hits that distant border, the tubulin network carries a chemical messenger directly into the nucleus in seconds. Once the messenger arrives, it actively alters which genes are expressed. The living organism is telling the genome what to do, not the reverse. This reactive network, anchored by the membrane, functions as the cell's true control system, allowing us to edit our code to survive immediate environmental threats. This ability to react and adapt introduces a biological concept called robustness. It defines how living physiological networks maintain their stability under intense pressure. This animation shows the mechanics of that resilience. If a specific gene or protein fails, the larger biological system doesn't immediately crash. It automatically reroutes the flow to compensate for the localized breakdown. Conditions like obesity, dementia, and heart disease emerge from the breakdown of these entire networks, involving thousands of interactions across the body. Our physiological networks are continuously reshaped by macroscopic external factors. Our diet, climate, and daily social interactions exert an epigenetic influence, actively rewiring our biology. The global scientific establishment is beginning to acknowledge this reality, shifting away from biology centered entirely on the gene. The future of medicine relies on understanding the architect, the living, breathing systems of the body, rather than just reading the passive ink of the DNA blueprint.